The invisible threat to crop production. Hello, my name is Vincent and I work as a plant pathologist at Agroscope, the agricultural research station of the Swiss government. I started to do applied research on the control of soil-borne diseases in 1992. I would like to share with you some of my experience, which I collected during this time. From the mountainous world of the Alps, I will move to a more maritime environment. The cruiser HMS Belfast in the centre of London is easily recognizable as a warship, quite threatening to the surrounding world. It is the same with airborne diseases. They are also easily to recognize already at the start of an infection. You surely know such diseases like powdery mildew, rust, anthracnose, fusarium head blight, and others. Next to the fact that airborne diseases are visible and relatively easy to identify, they also have the advantage for the producer to be easily accessible for treatments with fungicides or other plant protection products, which also means that control measures can be taken during the growing period of the crops. But there's another threat to the crops, often ignored, because it is, like a U-boat, most of the time invisible. This threat is caused by soil-borne pathogens and nematodes. Strawberry are quite susceptible to a range of pathogens in the soil, which cause soil-borne diseases like, for example, Verticillium-wilt. The Verticillium dahlia fungus survives in the soil as microsclerotia, a surviving structure. These tiny little dark-colored structures germinate when a root of strawberry or of another susceptible host plant is in proximity. The pathogen then enters in the roots, colonizes the vascular system of the plant, which leads to an obstruction of the water and sap flow in the plant. As a final result, the plant will wilt and, in the worst case, perish. But wilt and death of the strawberry are just symptoms for the disease in the final stage. You will also see this with other host plants of Verticillium, such as eggplant or sweet pepper, or with other diseases such as corky root or black dot disease of tomato. Wilt and death of plants are not a symptom limited to an infestation by a soil-borne pathogen or nematode. Other factors, such as too little or too much water, can also cause wilt, like in this pepper field, which was flooded after heavy rainfall. More precisely, 621 millimeters in 5 days. The pathogen itself is, even at the advanced stage of a soil-borne disease, nearly always invisible. Clearer symptoms than just wilting can be seen for some diseases inside the plant, like a darkening or browning of the vascular system that can be seen once you cut open a stem or a root. If you want to see other symptoms indicating infection by a soil-borne pathogen, you have to dig. Vascular diseases such as Verticillium or fusarium-wilt do not affect the roots on the outside, but other pathogens colonize and destroy the root systems. Once the roots are washed, some symptoms are easily recognized, such as the corky root disease. Others need a closer look under a magnifying glass, like the black dot disease caused by Calotrichum coccodus. In some cases, even though the crop is infected by a soil-borne disease, no symptoms on the above-ground part of the plants are visible. In this trial with strawberries, planted in a plot known to contain soil-borne pathogens, especially Verticillium, the soil on the left side was chemically disinfected before the planting, but not on the right side. Therefore, the plant on the right side had weaker growth than the ones on the left side. Even at the beginning of the harvest, no symptoms indicating a soil-borne disease could be detected. But there was a clear difference in yield, which was double in the disinfected soil, which means a soil without soil-borne pathogens. Another example are these rocket plants, infected by Pythium brassicum. The only but clear sign that there is a problem is the size of the plants. No symptoms on the leaves, but also not on the roots. In this case, the pathogen is inside the roots and disturbs their function in providing the plant with water and nutrients. Another point of the invisibility of soil-borne diseases is that the effect of the previous crop might be underestimated. I was testing short crop rotations during my thesis in Taiwan. The pathogen tested in this trial was Ralstonia solanacearum, causing bacterial wilt of tomato and many other crops. In this trial, I measured the population of the bacterium in the soil. The first crop grown were rice, cowpea and eggplant. The fourth treatment was a fallow, weed-free soil. The second crop was a susceptible tomato cultivar. With the exception of eggplant, which is also a host plant of Ralstonia, the population of the bacterium declined during the period of the first crop. Once the tomatoes were planted, the bacterium population increased again at different degrees, with the exception of the soil cropped to rice. Because the population of Ralstonia was quite high in the eggplant plots, the percentage of wilted tomato increased very fast, reaching 70% after 2 months' time. In contrast, tomatoes grown after the rice crop, with a very low bacterial population, had less than 10% wilted plants. There was also an impact on the yield, which was reduced by 50% for tomatoes grown after eggplant. A few years ago, we replaced the soil contaminated with several soil-borne pathogens of the glasshouses at our research station by a healthy field soil. For the continuation of the research on soil-borne diseases, we added a very small amount of the old contaminated soil to the new soil. The two major pathogens in the old soil were Colitrichum cocodes, causing black dot disease, and Parioncheta lycopersicii, causing corky root disease, also called brown root rot diseases of tomato. In 2009, 2010 and 2012, we conducted trials with tomatoes in the same greenhouse. At the end of each trial, we rated the disease symptoms on the roots. In only 3 years, the part of rotted roots went up from a few percentage to over 90%. When a non-grafted, susceptible tomato cultivar was planted. Tomatoes were planted every year, and the crops between two tomato crops were cucumber and lettuce. No soil disinfestation was undertaken. Actually, the solution consists in grafting on resistant rootstocks. The problem is that many rootstocks are not resistant to Colitrichum cocodes. So the conclusions until now are soil-borne pathogens are invisible, and therefore the presence in a soil is often ignored. Above-ground symptoms of diseases caused by soil-borne pathogens might not be recognized, especially at the early stage of the disease development. Clear symptoms of soil-borne diseases are hidden in the plant or below the ground. Infected plants without symptoms can maintain populations of soil-borne pathogens at a high level. And repeated cultivation of crops susceptible to soil-borne pathogens can increase their population to a high level within a few years. For this reason, a crop rotation with host plants and non-host plants for the most prevalent soil-borne diseases alternate is an excellent way to avoid two big problems in your crop production. The best for soil databases contain information on the host plant status of 20 field crops, 29 vegetables, and especially interesting, 21 green manure crops. And this for more than 130 pathogens and 30 nematodes. Back to the maritime surroundings. There is another threat, often visible only at the last moment: pirates. Even though on this ship hosting the Jolly Roger in the last moment might be difficult. In the world of crop production, pathogens transmitted with seeds, tubers or planting material are such a threat. The anthracnose of marshmallow is an example for this threat. The roots of these crops are used for the production of candies and are grown in Switzerland by organic farmers. 20 years ago, in the field of several farmers, plants started to wilt. The causal agent of this disease was identified as Colletotrichum malvarum. Because the disease started to appear in the field of several growers in the same time, the suspicion was a seed transmission of the pathogens. Different seed lots were tested, and especially the seeds of one of the lots was strongly contaminated with different pathogens, among them also Colletotrichum malvarum. Consequently, the contaminated seeds were discarded, and anthracnose was not a problem anymore in the marshmallow production. Seed transmitted diseases are not really soil-borne diseases, but they start the infection from the soil. And some seed transmitted pathogens, such as Verticillium albuatrum or Colletotrichum cocodes, can stay in the soil and infect thereby a hitherto disease-free soil. For these reasons, seed transmitted pathogens are also included in the Best for Soil databases on fungal pathogens. I hope after this presentation the invisible threat of soil-borne diseases is now less scary. Being aware of this danger is the first step for an intelligent and holistic plant protection management. Thank you for your attention.